Rapid method for enumeration of E. coli Biotype I. Prepare cereal 10-fold dilutions in peptone water of food. Aseptically transfer sterile cellulose acetate membranes to the surface or dried glutamate agar. Transfer duplicate 1 ml aliquots of each dilution to cellulose acetate membranes, using a sterile glass Drygalski spreader. Distribute the fluid over the entire membrane, except the periphery. Incubate plates 4H at 35 degrees Celsius, facilitate resuscitation. Transfer each membrane to tryptone bile agar plate and incubate for 18H at 44 degrees Celsius. Remove lids of plates. Add 3 ml Kovacs reagent into each lid. Remove membrane from agar and immerse in reagent for 5 minutes. Remove membrane and drain excess reagent. Dry the membranes under a UV lamp. Count pink-stained colonies within 30 minutes. Calculate the number of type I E. coli by selecting the dilution, giving an average of 20-50 pink colonies in both membranes, and multiply total by the dilution factor.